Macroalgae are loosely speaking those that can be seen with the naked eye. Most of them are classified as benthic, which is to say that they fasten themselves to the seabed. But there is no need to be so formal in talking about benthic macroalgae. Seaweeds are a diverse group of mostly photosynthetic algae found in marine and freshwater environments. Seaweeds are made up of a special combination of substances which are very different from the ones typically found in terrestrial plants and which allow them to play a distinctive role in human nutrition. Most notably, the mineral content of seaweeds is 10 times as great as that found in plants grown in soil. As a consequence, people who regularly eat seaweeds seldom suffer from mineral deficiencies. Seaweeds are classified into 3 types: brown seaweed, green seaweed, and red seaweed. All 3 groups contain chlorophyll granules. Our focus is brown seaweed. Brown seaweed, which is known as bladder wrack, is a salty sea vegetable that's popular in many cuisines. Brown seaweed is alga that grows in cool oceans around the globe. You may have seen patches of its slimy green-brown branches washed up on the beach. Brown seaweed is a common ingredient in many Asian cuisines. People eat it raw, cooked, or pickled. If it's raw or pickled, it has a crisp crunch. Cooks serve up brown seaweed in a Korean soup called myeokguk. You can also incorporate it into plain miso soup or seaweed salad. Its texture and flavor can make bland foods more interesting. Brown seaweed is beneficial in cancer prevention and treatment, weight loss, diabetes, cognitive improvement, arthritis, asthma, and heart disease. Our main focus is to discuss its anti-tumor activity that is due to one of its active ingredients, fucoidan. Cancers are multifactorial diseases of various etiologies. They arise largely as a result of acquired genetic changes that alter cell function, leading neoplastic cells to gain survival or growth advantages. For cancer cells to survive, the generation of new blood vessels is required. Cancer leads to death mostly through tumor cells spread to distal organs called metastasis. Various pathways are disrupted in tumor development, which result from unbalanced programmed cell death, disordered signaling pathways, angiogenesis, and poor immune response against cancer. Most of the chemotherapeutic agents used in cancer treatment target these major deregulated pathways. Brown seaweed contains an element called fucoidan, which researchers have isolated and tested for its effect on cancer. According to recent studies, fucoidan has shown promise in eradicating or slowing the spread of colorectal and breast cancers. Fucoidan is a sulfated polysaccharide found in brown seaweeds. The brown seaweeds containing fucoidan are widely consumed as part of the normal diet in East Asia, particularly Japan, China, and Korea. Fucoidan is non-toxic, biodegradable and biocompatible compound approved by Food and Drug Administration (FDA), as generally recognized as safe GRA category as food ingredient. Fucoidan plays a vital role as anti-inflammatory, anticoagulant, prevents blood clotting, antithrombotic, reduces the formation of clots, anti-angiogenic, reduces the growth of new blood vessels, antiviral, anti-tumor, anti-inflammatory, anticoagulant, prevents blood clotting, antithrombotic, reduces the formation of clots, anti-angiogenic, reduces the growth of new blood vessels, antiviral, anti-tumor, antioxidant. Fucoidan mediates its activity to fight against cancer through various 1. Induction of cell cycle arrest 2. Fucoidan promoting the apoptosis pathway 3. Inhibits the new vessels formation 4. Decreased tumor cells metastasis or spread 5. Fucoidan regulates the signaling pathway 6. Immune system activation 7. Inhibition of malignant transformation. It induces cell cycle arrest in G0 and G1 phases. It has a special role in chemo-resistant bronchopulmonary carcinoma by inhibiting its cell cycle at G1 phase. It is suggested that fucoidan significantly down-regulates the cyclin D1, cyclin D2 and CDK4 in cancer cells and can be useful in lungs and bladder cancers. The second one is fucoidan promoting the apoptosis pathway. Apoptosis is programmed cell death, which is a physiological process to maintain body homeostasis. It is necessary in terms to prevent cancer by removing unwanted cells through a programmed cell death. Several studies examining a variety of cancers such as blood, lung, breast and colon cancers have shown that fucoidan-mediated cell death occurs through triggering apoptosis to prevent the body from these cancers. Next one is fucoidan inhibits the formation of new vessels by which tumor cells receive their oxygen and required nutrients. Fucoidan has been found to inhibit the binding of VEGF, a key angiogenesis-promoting molecule, to its cell membrane receptor. 4. Fucoidan significantly decreases tumor cells' metastasis to the lungs. It was first reported that fucoidan inhibits cell invasion through competing with tumor cell binding with basement membrane. Subsequent studies then revealed that fucoidan binds to fibronectin with high affinity and prevents attachment of tumor cells. Fucoidan reduces the spread of human breast adenocarcinoma cells plated on a surface containing fibronectin. Subsequent studies then revealed that fucoidan binds to fibronectin with high affinity and prevents attachment of tumor cells. Tumor invasion requires the secretion of proteolytic enzymes by tumor cells to break down the extracellular matrix proteins. Fucoidan attenuates both expression and activity of these enzymes. 5. Fucoidan regulates the signaling pathways. The extracellular signal-regulated kinase pathway is often hyperphosphorylated and upregulated in a variety of human cancers. The potential for developing anti-cancer agents that cause ERK's dephosphorylation and pathway blockade have been explored by using fucoidan. 6. Fucoidan activates the immune system. Fucoidan increases both activity and number of natural killer cells. It increases the number of cytotoxic T cells for specific lysis of breast cancer cells. 7. Fucoidan inhibits malignant transformation. Fucoidan inhibits neoplastic transformation of malignant cells. Transforming growth factor is believed to promote tumor development and metastasis, such as during late stages of breast cancer development. Fucoidan decreased the expression of TGFRs and affected the downstream signaling molecules. Epidermal growth factor is another carcinogenesis promoter, which induces tumor transformation through overexpression and activation of EGF receptor. Fucoidan markedly reduced the EGF receptor activation through hypophosphorylation. It also inhibits EGF tumorigenic activity. The goal of cancer treatment is eradication of tumor cells, ideally with minimal damage to healthy tissues. Because of the side effects of many current treatments, the use of natural substances of low toxicity is of interest. Fucoidan contains strong anti-cancer bioactivity. Since fucoidan also possesses immunomodulatory effects, it is postulated that it may have protective effects against development of side effects when it is co-administered with chemotherapeutic agents and radiation.